Dr. Siddhartha Mukherjee is a 54-year-old hematologist and oncologist. Since 2009, he has served on the faculty of Columbia University, where he is an associate professor of medicine and a practicing physician at New York Presbyterian Hospital. His passion for cancer research is driven by the hardship of watching his patients struggle while fighting cancer. Mukherjee and his team at Columbia research the biology of normal and malignant blood development, focusing on diseases such as leukemia. He was born in New Delhi, India, where he lived his entire life before moving to the USA to attend Stanford University, where he graduated as a Rhodes Scholar with a Bachelor of Science degree in biology. He then went on to get his Doctor of Philosophy in Immunology at Oxford University and attended medical school at Harvard University. He completed his internal medicine residency and an oncology fellowship at Massachusetts General Hospital and is board certified in medical oncology. When Dr. Mukherjee is not working as a doctor or researching, he is writing. He is a science writer and has written 4 successful books. His most popular book, The Emperor of All Maladies, A Biography of Cancer, was a winner of the 2011 Pulitzer Prize in General Nonfiction and a #1 New York Times bestseller. I was intrigued by Dr. Mukherjee and his work when we watched a video in class during Cell Week, and he explained how CAR T cells work to fight against a young girl's relapsing cancer. After further researching him and his work, I admire the person he is and how he cares for his patients. I identify with him because I think he is someone who genuinely cares about his patients and making a difference in any way possible, and I have always been a really caring person who wants to help people in any way I can as well. He works as a hematologist and oncologist, and his research lab studies the biology of blood development, specifically diseases such as acute myelogenous leukemia, otherwise known as AML, and his eventual goal is to develop novel drugs against these diseases. He is focused on the links between normal stem cells and cancer cells, as he believes that the biggest problem with current cancer drugs is that they are lousy at distinguishing between cancer cells and healthy cells. Siddhartha is an author on a paper that breaks down an experiment where he and other researchers aim to assess whether gene editing of HSPCs to delete the CD33 gene could protect normal hematopoietic cells from CD33 targeted immunotherapy, thereby allowing selective elimination of CD33 positive leukemia cells. This figure represents data on the effectiveness of CD33 targeted therapy in CAR T cell treatment in an AML mouse model. Here's a breakdown of the panels. Panel A, human AML cells are injected into mice. The mice receive different treatments including PBS, CAR33 T cells, anti-CD33 therapy, or a combination of CAR33 plus GO. The disease progression is monitored at multiple time points. In Panel B, flow cytometry plots show the percentage of AML cells in the bone marrow across different treatment groups. The control groups show high AML levels, while anti-CD33 therapy and CAR33 plus GO show significant reductions in AML burden. In Panel C, there's a scatter plot showing total AML cell burden in the bone marrow. The AML levels are significantly lower in CAR33 and GO treated groups compared to the controls. In Panel D, there's in vivo imaging of leukemic burden across different treatment groups. The control groups show widespread leukemia, while CAR33 treated mice show reduced signals. In Panel E, there's further imaging of leukemia progression. The CAR33 plus GO group shows the most significant reduction in disease burden. Panel F quantifies human CD45 plus cells, which are indicative of leukemia in peripheral blood over time. The CAR33 and GO treated group shows sustained reductions in leukemia compared to the controls. A couple of years ago, Mukherjee and his Columbia research collaborators achieved a milestone in a series of experiments where they were able to completely eliminate AML from sick mice by simultaneously giving them Mylotarg, a chemotherapy drug, and infusions of stem cells that they had genetically altered to produce myeloid cells that are invisible to Mylotarg. The experiments, which followed years of laboratory preparation, sent tons of excitement through the biotechnology and pharmaceutical communities, and Mukherjee helped launch a startup called Vor Biopharma, with the goal of one day bringing his team's therapy to hospitals and clinics. His company is planning to initiate a clinical trial that will give Mylotarg in tandem with Vor's therapy to AML patients who have failed to respond to traditional chemotherapy. Siddhartha Mukherjee is an incredibly passionate, intelligent, and inspiring doctor and researcher and writer who continues to break through barriers with his cancer research. Hopefully, through his work and work to come in the future, we can find a way to cure various types of cancer. Thank you for watching.